Dear students, a very warm welcome to the today's session on biology classes. So today we will be dealing with the chapter of cell, the basic unit of life. So first let us see what is the scope of syllabus as prescribed by the NCERT. So this is the basic unit, that is unit 3, cell structure and function. So under this unit we will be looking into the first chapter, that is chapter cell. This is chapter 8. That is cell, the basic unit of life. So in this chapter, as prescribed by the NCERT, so we will be dealing with the definition of the cell theory and the contribution of the scientists, and then we are going to look at the two different types of the cell, that will be the prokaryotic cell and the eukaryotic cell, and then we will also touch upon the concept of the plant cell and the animal cell along with the diagram and the differences, and then we will also look into the various organelles that is present within the cell, and we will dwell deeper into the structure of nucleus, the nuclear membrane, and also the chromatin network. So for the PUC board exam, this chapter carries a weightage of total 8 marks. So this is for the PU board exam. So roughly about 12% of your total marking. So in this you can expect one question for part A, that is going to be a 1 marker. Then you can expect one question for part B, which is going to be for 2 marks, and last you can expect one question for part D1 or D2, which is going to be for 5 marks. As per the trend, we are observing that in K.8 and NEET, this chapter we are going to get either 1 or 2 questions. So that is about the scope of the syllabus, the marking scheme as per the DPUE examination guidelines, and then what you can expect for the Karnataka CET exam and then the NEET exam. So this is going to be the fundamental section or the plan of how we are going to deal with this chapter. So now let us begin with this chapter. So now I am sure you all have seen or read in chemistry about electron, proton, neutron. Yes, so they are all the components which form the smallest or the basic structure of matter, that is atom. So similarly, like how two atoms are going to combine to form a molecule, and then molecules will aggregate together to form compounds and then higher molecular weight compounds. Similarly, in the living world or in the living domain, you have the smallest and the simplest structure, which is the structural and functional unit of life, and that is what we call as cell. So like how atom becomes the fundamental unit of matter, similarly cell becomes the basic structural and functional unit of life. Now the next question arises, how am I so sure that cell is the only structure or the fundamental structure? So here you have to pay attention to the contribution of scientists. So first, we have the scientist Robert Hooke. So he was the scientist who studied about dead cells. He was examining a section of cork under the microscope, and he found compactly arranged compartment-like structures that was resembling the honeycomb. So Robert Hooke was observing or examining a section of cork under the microscope, and he was the first person to study about the cells. He studied about dead cells. So what he observed was, he observed a pattern or arrangement which had compact arrangement, okay. It looked like closed compartment like structures. It resembled the honeycomb shape, and that is when he called them as cellula. So Robert Hooke was the first scientist to observe the dead cells under the microscope, and he coined the term cellula, which means cells. Therefore, Robert Hooke is rightly labeled as the father of cell biology. I have told you that cell is the fundamental unit of living form, but then again this is dead. So now comes the contribution of another scientist, Anton van Leeuwenhoek. So he was the first person to observe living cells as he was examining So Anton van Leeuwenhoek was observing samples from the pond water, and when he was looking at the pond water sample of the microscope, he for the first time observed microorganisms which were alive. So therefore, Anton van Leeuwenhoek is called as the father of microbiology. So in this chapter, the first scientist that you ought to remember, one would be Robert Hooke and the other would be Anton van Leeuwenhoek. So these were very generalized descriptions or discoveries regarding the fundamental structural and functional unit of life. Now we have to move to something more concrete which will help us understand what is the cell that we are talking about, what is it that it is made of, and what is a property of the cell that enables it to be called as the basic structural and functional unit of life. So next we will take up the subheading of cell theory. So next heading we will deal with this cell theory. I am sure you have studied so many theories in your different subjects. So this is a very vital theory which is going to explain about these cells. So let us see who are the scientists who contributed to this. First we have Matthias Schleiden. He is a German botanist. He in the year 1838 studied various plants, because botany is a branch of biology that deals with plants. So Matthias Schleiden in the year 1838 he studied about various plants, and he found that each plant is made up of different types of cells, and that is going to form the plant tissues. In the next year we have Theodore Schwann. He is a British zoologist. So zoology is a branch of biology that is going to deal with the study of animals. So in the year 1839, he studied various animal cells and he observed that the limiting membrane in the animal cells is very thin, and now it is known as the plasma membrane. In the next few years, both these scientists, Schleiden and Schwann, exchanged their results so that they could come to a common conclusion. So that is when they came to a conclusion that plants and animals are both made of cells, but these cells are different from each other, in the sense that Schwann proposed that in the animal cells the limiting membrane is very thin, and Schleiden observed that the characteristic feature of a plant cell is the presence of a thick, dead, rigid cell wall. Next, we have the contribution of the third scientist. He is Russian. His name is Rudolf Virchow. Okay, so he proposed a concept called as omnis cellula e cellula. So these two proved beyond doubt, the two scientists Theodore Schwann and Matthias Schleiden proved beyond doubt that all living organisms are made of cells and they are going to be different from each other when compared to plants and animals. Now the question was, how is it that there are so many cells in the body, and how is it that the dead and worn out cells are replaced by the new ones? So that was explained by Rudolf Virchow in the year 1855. He proposed what is known as omnis cellula e cellula. So from the discoveries and studies of Robert Hooke, we know that cellula means cells. You all would have heard the statement, God is omnipresent, or crow is an omnivorous animal. So omni means everything or everywhere. So here omni could mean every. So Rudolf Virchow proposed that every cell is going to arise from a pre-existing cell. Every cell is going to arise from a pre-existing cell, and the common ground for all these three scientists, all of them came together and they proposed what is known as the modern cell theory. Matthias Schleiden, Theodore Schwann and Rudolf Virchow put forth what is known as the modern cell theory, and it is going to have four postulates. So it says that cell is the basic structural and functional unit of life. Cell is the basic structural and functional unit of life. Next, all organisms are composed of cell and cell products. Next, we take into account the contribution by Rudolf Virchow, which states that cells arise from pre-existing cells, which is known as omnis cellula e cellula. So this is about the postulates of the modern cell theory. So now so whenever there is a theory, obviously there will be some exceptions. So let us now look at the exceptions. So what could be the exceptions to the cell theory? So exceptions will be that virus is acellular and lacks metabolism. So viruses, they are acellular, that means they do not have a cellular structure and they do not perform any metabolic functions. Next, prokaryotes. So pro meaning primitive or the very initially formed, very basic level organisms. Karyon refers to nucleus. So prokaryotes are those organisms which have a very primitive nuclear organization. So these prokaryotes, they lack nucleus and organelles. They lack the nucleus and then the organelles. So these are the very important exceptions. The other one would be a enucleated cell. Which is going to be a at maturity, the mammalian red blood cell does not possess the nucleus, but still it is going to obey the cell postulates of the cell theory. So that is why this is an exception. So that would be the theory that we already discussed, and these are the exceptions to the rule. So now let us deal with the attributes of the cell. So what could be the attributes of the cell? So attributes will refer to the characteristics. So basic characteristics. So here we will talk of shape, size and number. So we will be dealing with the shape, size and number. So first one, let us talk of number. So when an organism is small, it's going to have lesser number of cells. As the organism grows bigger in size, due to the concept of cell division, the organism will have more number of cells. So based on number, you are going to have unicellular organisms. Unicellular meaning they are going to be made up of only one cell. So that will be example algae, Chlamydomonas. Next, as opposed to the unicellular organism, we are next going to have the multicellular organism. So multi meaning many. So they are the organisms which have more than 2 cells. Okay, so here it can be mammals, birds. So that is the attributes of the cell based on number. So next let us see the shape of the cell. So now we have already seen the number. Now let us look at the shape of the cell. Now See, in our human race, if you take for an example, some people are tall, some people are short, some people are fat, some people are lean. So there are differences in how the body is going to be built. So here also, the cells, all of them are not going to have a same single or a uniform shape. The shape is going to determine what function they are going to perform, or you can even call it vice versa. The shape determines the function, and also the function determines the shape of the cell. So the cells can assume different varieties of shapes, and that is going to depend on the function that it is going to perform. So here the shapes of the cell, they can be discoid, they can be spindle shaped, they can be cuboidal, they can be branched, they can be cylindrical. So in the discoid, best example is the red blood cell. So if it's the shape of a disc, it is going to have more surface area, and there is going to be better exchange of oxygen and carbon dioxide. Next, spindle shape will be the smooth muscle cell. So this is the involuntary muscle that is present lining the esophagus. Next, cuboidal. This is the epithelial cell. So this is going to be present in the kidney or in the nephron, and this is going to help in reabsorption. Next, we have branched. So this is going to be the cardiac tissue or the cardiac cell. Okay, it is going to show branching so that it's easy for it to conduct the nerve impulse along the entire structure of the heart. Last, we are going to have the cylindrical or the tubular structure. The best example is going to be the nerve cell. It has to be like a cylinder or a tube because it has to connect the central nervous system, that is the brain, to the periphery, that is the finger. See, now if I have to hold the chalk, I have to receive the impulse from the brain and the spinal cord to tell it how much pressure I have to exert so that I just hold the chalk and not break it or make it into a powder. So that is why the shape of the cell is a very important characteristic, and based on what function it can perform, these cells are going to assume variety of shapes. Now, the most interesting concept is going to be the size of the cell. The most important concept is going to be the size of the cell. This is actually a very interesting concept because here we now understand what could be the shape, what could be the size of the cell and what is the smallest size it can attain. Now, as we all know, the cells can be microscopic, that is not visible to the eye. They are going to be visible only in the under the microscope. So they are going to range from few micrometers, that is 10 to the power 6 meters to a few meters. Yes. So the cells can vary from micrometer all the way up to a few meters. Now let us see certain examples. What could be the smallest cell? The smallest cell is mycoplasma. It belongs to the pleuropneumonia-like organisms, or what we call as PPLO. Now, coming to the humans, what could be the smallest cell in the human? That is the human sperm cell. Next, largest cell in the animal kingdom would be the ostrich egg cell. Ostrich egg cell, and as opposed to this in the human, it is going to be the human egg cell or the human ovum. Now we are going to look at the longest cell. In plant kingdom, we have acetabularia. This is the longest cell. This can grow anywhere up to 6 to 10 centimeters. And in the human, it is the neuron. It is the sciatic neuron. Okay. So this is going to extend all the way from the lower back to the lower limbs. So this is very important to remember because this can come as a 1 mark question or it can appear in the NEET. So this is very important. Fine. Now as I have explained, we have to look at another concept in this size which is called a surface area and the volume ratio. So the next concept will be surface area and volume ratio. So now what I would do is, we can assume a cell to be cubical in shape. Okay. Now we are going to have cell A where the side is 1 centimeter and then another cell, cell B, where the side is 10 centimeters. Now, what could be the surface area of the cube? The surface area of the cube is given by the formula 6a square. Is given by the formula 6a square. So that will be 6 into 1 into 1, which will be 6 centimeter square. If I take up for cell B, it will be 6 into 10 into 10, that is going to be 600 centimeter square. Now the volume of the cube is given by the side cube. So here the side is of 1 centimeter, so it is going to be 1 centimeter cube, and here it is going to be 1000 centimeter cube. When I perform surface area is to volume ratio, I am going to get 6:1, and here I am going to get 0.6:1. So here for cell A, it is just 6 divided by 1, which is 6:1, and here it is 600 divided by 1000, that is 0.6:1. So with this, where have we arrived? What are we going to conclude out of this? So here, whenever the cell is going to have a higher surface area is to volume ratio, whenever a cell is going to have a higher surface area to volume ratio, it is going to be efficient in the surface transport. Phenomena such as whenever the cell has more surface that is exposed, so as a result there is a better exchange. So therefore the cell is going to be more efficient in the exchange process, and also it's going to be more efficient in performing its functions. Reason being, it's going to get its nutrients and at the same time it's going to expel the waste out of the cell. The second and the most important postulate. Now imagine this cell B is this big, then the cell A is this small. Though being this small, it's going to have a higher surface area to volume ratio and is better efficient than a bigger cell. So now the question is, will you prefer a single large cell or would you like an aggregation of the small cells? So if I have a single large cell, the total surface area volume ratio is going to be 0.6:1. Now since I am going to have the same volume, say the volume is 1000 centimeter cube, I can have 1000 cells with side 1 centimeter, and each cell is going to have the surface area volume ratio of 6:1. So therefore, it is always preferable to have a group of cells rather than a single large cell of same volume. That is very important, because the volumes are different, then obviously the ratios are going to change. So from this surface area volume concept, what we understand is higher the SA and V ratio, better is the efficiency, and therefore the cell is going to have enhanced function. It's always preferred to have a group of cells rather than a single large cell. Both should be of the same volume. So with this, we are going to complete about the attributes of the cell.